So we can continue on with the next invited talk that we have. Our next speaker is Mireya Plass. She's a group leader at the Bellvitge Biomedical Research Institute in Barcelona, Spain. And her group is working in applying single-cell method to study gene regulation, and particularly post-functional modifications, and mostly focusing on neurodegenerative diseases. So, Mireya.
Thank you very much. Oh, I didn't figure out how to change slides. I'll go that this thing works. Okay, so hi all, and thanks first of all for inviting me. I think it's always nice when people, take you to a different country and a different city that you have never visited, and you can learn a bit about what they are doing. So today I will try, so I don't know where to start. Today we'll try to tell you about the work that we are doing in the lab. And basically right now we are developing a method that is called Scalpel. That is a tool for isoform quantification at the single cell level. And because I don't know really the background of anybody, so I will try to explain all the words from the title, and maybe we can take it from there and everybody understands a bit. Should this is way better, right? Great. Okay, so just before starting, I just want to say that the work that I'm going to be presenting today has been mainly the work from France and also a PhD student in the lab that has developed the computational tool, and also with the help from Anna, that was the person generating the data. So I think that those two things are both important. Okay, so let's start. Just in case that you don't know, it's very easy to explain what is single-cell transcriptomics. So you have to think about it as a new method that gives us like a new resolution for working with NGS data, right? So until now, let's say we were here, right? But we can have with both RNA-seq or bulk copy technology, proteomics, chipset, whatever. So basically, we were at the level of systems and organs. So we could take, for instance, a tissue and characterize it, see how it's different from a different tissue, or we could get a cohort of patients and compare whether the case and the controls have particular mutations or have changes in the transcriptomic profile. And in the end, get the cell population averages, right? So we could have a cell culture and see what's the expression profile of this tissue. But we were just there, right? So just everything is bulk, so we can never go to the single cell level. And that's what we get with the single cell transcriptomics. So we go to the cellular resolution. So you could imagine that instead of seeing the human body like this, we see it like this. So we can see all the cells that compose this organism. And now what we can do is like much cooler things, of course. For instance, we can take all the cells and we can try to identify all the cell types and states. So it's not only about having muscle cells, but also muscle cells during regeneration, or for instance, as Maria was saying, in our case, trying to see what happens in the neurons in neurodegenerative diseases. And we can study the composition of these organs and tissues, and we can also see how gene expression is constant and variable within each of these cell populations. Not only that, but we can go further than that and try to understand the further lineage relationships, so try to understand how these different cells are related to each other. Whether we can have a switch that is activating transcription and it's making one cell convert to another one, or how, cells relate, for instance, the stem cells relate with different cell types, and what are the gene programs driving these differences. So I hope that this would enough for single cell, no? So we can continue. But then it's like, how can we get the single cell resolution? And I would say that for the last, now almost 15 years, a bunch of methods have been developed. So this slide is not so updated, but I think it's still the nicest that we can find. And what you can see is like, in 10 years, basically, we have moved from people that were sequencing a single cell manually to get the genes that the cell was expressing, to doing an experiment that takes you a few hours, and then you can sequence tens of thousands or hundreds of thousands of cells in a single experiment. And this not only is that, but we can also get much more information. So we can get maybe 10,000 genes in each of the cells, sometimes 15,000, depending on the technology you use. I would say that the most common technologies as of today are these microfluidic, this one, nanodroplets. That basically, I will explain in a second what it is. So the idea of this technology is that basically we can start from any tissue that we have. And as long as we can associate all the cells, we can get their transcriptomic profile without doing anything else. And the main idea is that we are using a microfluidics device. There are different technologies using this, but the principle of how they work is always the same. And basically the idea is like, you would have your cells that you obtain from your cell culture, from your favorite organ, or from anywhere you can find them. And you're going to encapsulate them in micro droplets together with these beads. And actually these beads are the key, right? Because if we zoom in one of these droplets, basically what we have is this. So once we get the cell inside, the cell is lysed and it releases all the RNAs that it has. And then these beads contain tags that are going to allow us to mark like all the RNAs from a cell with the same identifier so that afterwards when we sequence we can know what it is. And we not only mark like the cell but also the mRNAs itself. So we can know how many RNA molecules a cell has and which are all the RNA molecules that come from a particular cell. So basically that's the summary, right? So when we sequence, we'll get a piece of read that is coming from the genes of interest that we have, and the other piece that will give us this information of the UMI that tell us about the RNA molecule, and then the cell barcode that tells us about which cell they come from. And we end up having something like this, digital gene expression matrix. That is very simple. It's a plain text file where you have basically all the genes from your organism with all the cells, and then you can count how many times you see each of the genes. And this is basically what we do. So, but thanks to the single cell transcriptomics, these cells are actually not like this, as what we were saying in bulk omics. So they are all of them different, and then, as I was saying before, we can compare not only the organs, but we can compare the proportions of cell types, the RNAs these cell types express. There are even some technologies where you can get the proteins that these cells express, or the chromatin. And now we have a much more richer picture of how the organs are formed. So, and what can we use this single cell transcriptomics for? So, I have to say that people have used it for almost anything that you can think of. So, for instance, there has been this very lovely picture that it looks like a map from the 15th century, but in reality, it's like a map of the neurons that mouse have during development. Right? And then you have different populations of neurons, different types of cells. You can also use it not to make also a very deep atlas, but you can use it to infer the relationship between cells. Like for instance, in this paper, we studied how stem cells give rise to all the cell types from an organism and identify the genes that are driving these cases. But you can also use it for medical applications, like for instance, people are using single-cell transcriptomics to understand the heterogeneity within cancers, in tumors, and even to see how cells change, for instance, after chemotherapy. Or finally, also like technological applications. For instance, to develop CRISPR screens. So the idea that to prove the function of thousands of genes simultaneously, because now you can use each cell as a reaction chamber, and you can record everything that is happening there. So this is actually awesome, and if I don't think about that we would be doing this, 15 years ago, I wouldn't have believed it. But the question is that all these technologies, they have a key missing part, right? And that's actually what we are trying to solve. And the missing part is that they measure the expression of genes disregarding the diversity that these genes generate. So what do I mean? So let's imagine this is a gene, right? And in classic biology, we'll have this idea that you have a gene, and this gene gives rise to a protein, right? And here we have some RNA and everything. But the reality is that from one gene, we don't get one only version, so that the genes produce different versions of transcripts, right? That have changes in the beginning and the end, they even can have missing pieces in between, and this has consequences for the proteins that we generate in the end, for where they are located, the functions that they do, or even, I don't know, the activation in case they are enzymes and things like that. And all this is missing in current single cell works, or in most of them. So, and why do we have this diversity? It's because of RNA processing, that is what we are trying to study in my lab, right? So here would be like a classic picture of what happens to an RNA since you transcribe it from the DNA, then you generate like proteins, I think it's this here. And here in black, what you have is all the processing mechanisms that exist within the cell and that actually are affecting which mRNA, so which versions of the genes you are expressing. Among all these different steps, as you can see, there are plenty. In my lab, we're interested in studying this one, that is polyadenylation. So polyadenylation is the mechanism by which we define the ends of the gene. So you see it's like a very specific process, it doesn't do much. So, but basically when you have your gene, you define whether it finished here or there, like a bit longer, a bit shorter. In most cases, so this is what it's being depicted here. And in most cases, these changes only affect to the non-coding region. So it's actually a piece of the gene that doesn't make proteins. So you could say maybe it's not so relevant, but the reality is that this region here contains binding sites for lots of regulatory elements. So other proteins, other RNAs that are going to be interfering with the function of this gene, and it's going to affect what happens within the cell. So I don't know what's, so I don't know which slide is here. So, but how can we measure this 3' isoform diversity in single cells? So as I told you before, well, single cell is ideal for this. Why? So as I just told you, we are trying to look at alternative coagulation that affect the end of genes, right? As I showed you before, single cell technologies, that is the most popular ones, are capturing the RNAs using these oligo-DT sequences. That what they do is they actually attach here to the end of the genes. So basically, when we are sequencing, our reads are not normally distributed in a gene, but they are mostly concentrated at the end. So this is like actual real data from the 3 main technologies that use this Droplet base and are the most popular, especially 10x is the most popular ones. And you can see that most of your reads are really, really close to the end. Therefore, we have a technology that is ideal for doing this thing that we are interested in, although no one is doing it. So, yeah, it's great for doing accurate quantification of 3' ends. But how can we do it? So for that, we have developed the Scalpel. So it looks like a metro map from London or something like that. But the main idea is that we can go from a reference annotation, so a set of transcripts or isoforms that you want to measure, and your single cell data. And now what we can do is to generate also a quantification of the single cell data, but we are going to decompose the expression of the genes in the expression of different isoforms. And how do we do this? I hope that you all understand this already from this slide, right? So let's try to go a bit step by step to try to see if we understand a bit how we do it. So there are like 3 main steps in Scalpel. The first step would be to define offset of transcripts to quantify. And what do I mean with this? So let's imagine that we have this example. This is actually a real case of IF1 gene. In human contains 3, 4, 5, 6, 7 different versions. And as you can see, as I was telling you in the example, they change in the end, in the beginning, they have missing pieces in between. But what we know is that in human cells, usually we don't use all these versions. We only use a subset of them. Some of them are important for particular cell types or for particular tissues. So, but how can we know which ones are important? So how can we choose? For this, we did like a trick. And we use Salmon. So Salmon is a program that was developed by very clever people in the States. And basically the idea is that it's going to try to quantify your transcript isoforms using bulk RNA-seq. But in the end, we have a bunch of reads, so we can treat these reads as if they were like a normal RNA-seq experiment. And basically what we can do is like, we make an index of our transcripts that basically consists of making like k-mers of all of them. And then we take our reads and we break them in k-mers too. And then basically we see how well the reads from our reads map to the transcripts, and then we quantify this. Okay, it sounds a bit, I don't know if you are familiar with this, but the result is, it's like mapping, but it's way faster because you're really not comparing the sequences, but you just have an index of abundances of k-mers associated with different features, and in this case RNA transcripts. So once we do this, what happens is that this set of reads allow us to quantify our transcripts, and then we can see that based on where they are distributed in the transcript, we have some isoforms that are like really abundant, some others that are basically not expressed. So we can get rid of this. Why? Because in this way, what we are going to be able to do is save a lot of time computing things that actually are not useful for anything. But the other problem that we have when we have done this is that we have genes that have the same ends. You see, for instance, here. And as I showed you before, our reads are only in the end. So the technology that we are using will never allow us to distinguish between these and these because we are not going to have enough coverage. So we are using, in this case, this data to get rid of the isoforms that are less frequent among all the ones that are kind of identical in the end, right? So for instance, these two. And then we discard the one that is less used, we keep the one that is more used. And with this, basically, we end up with a set of transcripts that are more or less abundant, but we can use for the quantification. So this was the first step of Scalpel. So now what do we do in the second step? We clean the data. Why? Because single-cell transcriptomics is great, but it's very noisy, and you have lots of artifacts that we know. The biggest of these artifacts is what we call internal priming. That is the fact that because we are capturing the mRNAs with an oligo T, this is going to give a signal anywhere where we have A's that is the same that we have at the end of the gene. So we need to get rid of this signal in order to quantify correctly alternative polyadenylation sites. And also we want to get rid of reads that map in introns. So in case that you are not familiar, basically introns are the pieces of the genes that are removed when the mRNA is being processed. And the problem is like if we keep them, they are going to look like they come from like mature mRNAs, but they are not, and they will also interfere with our results. So this is what I was telling you about internal priming, right? So the normal RNAs like this would be A's, and this is going to make that the reads are upstream, but any region within the gene that also has a bunch of A's in the sequence would also generate reads upstream. And this is going to give us a signal that is wrong because this doesn't correspond to any gene end. So we try to remove these internal priming sites. So how do we do it? Yeah, maybe now it gets more complicated, sorry. So we have the same example as before, our gene, and these are our reads. But as I told you before, these reads are marked with a unique molecular identifier that identifies the mRNA molecules where they come from. And surprisingly, what happens is that you would expect that you get one read from each of the mRNAs, but it's not true, because you get several reads coming from the same mRNA. And then we need to consider this into account, for instance, when we are discarding these internal priming sites, because although maybe these reads are here and these reads are here, I know they all come from the same mRNA molecule, so if one is noise, all of them are noise. I don't know if I explained myself, otherwise you can ask. So this is the idea, right? So we have internal priming sites, we find the reads that are just upstream, and then we discard them. And then we are done. But all this is great, but I still haven't shown you how we can quantify anything, right? So that's the last part of the program. How we can take all this data that we have cleaned with this annotation and extract, so decompose the gene expression data into expression of the different isoforms. So in this case, we are doing something very simple, that is basically running an EM algorithm. And just because I don't know whether you know what it is, so basically it's like an algorithm that is iterative, in which basically we try to predict something based on the observations that we have. In our case, what we are going to try to do is calculate the probability of each of these isoforms given the reads that we are seeing, right? Well, the fragments, this combination of reads that come from the same mRNA molecule. And for that, what we do is we model the distribution of the reads relative to the three fragments. I have been showing you several times that the reads accumulate at the end, so we can use this information to figure out where the reads are supposed to be coming from. And yeah, this to show that instead of using the empiric probability, we are trying to fit a parametric distribution so that the data is a bit less noisy. So once we have this, that is what is going to be driving how we select the isoforms. The other thing that we have is that we have to define a transcript relative abundance, and it doesn't matter, apart from zero, it doesn't matter much which number you put, it would give you always the same result, and just for easiness, it's like 1 divided the number of transcripts to quantify. And here what we are doing is to adding a weight, that is the transcript Salmon probability. As I showed you before, the Salmon, we have like a quantification of each of the isoforms. And because we know that some transcripts are more probable than others, we want to use this information to kind of modify how we assign the reads. And it's not only considering their position in the mRNA, but also like the abundance of the transcript itself. And when you do this, you kind of repeat it again, you go to the end step, that basically what you do is to try to calculate the transcript relative abundance, that is this number that before it was like, for example, we would have 5 transcripts would be 0.2, and you do it considering the fact that you have already quantified the probability of the isoforms independently. And then you repeat these steps like a bunch of times, and in the end, magically, it converges and you can get your probabilities. And what do you do with that? So basically it means that we have gone from here to here. So now we have, instead for having one gene for each of the genes, we have the isoforms, and we are distributing the reads that we had before across these isoforms for all the cells. Okay, so I explained you everything that we have done. And now the question is, why do we care about this? What can we use? And on top of that, how can we know that this is working? Because I can tell you anything and maybe nothing makes sense. So we decided to set an experimental setup to validate whether this program is working. And for that, what we did was to make a very simple example. So we took 2 cell populations, fairly pure, that we know that they are different, and we know that on top of being different, they have changes in this 3' selection, so in alternative polyadenylation, and we did single cell with them. So that now what we can do is use this data to, for instance, quantify gene expression, like what everybody is doing, quantify isoform expression with the scalpel, and finally to see if we are able to identify the changes that we know that should be there. So the first step that we can do when you do single cell transcriptomics is try to see at least whether quantifying isoforms we are as good as quantifying genes for defining cell populations. And of course, if I say this, because the answer is yes, right? So this is going to be an example of a UMAP plot. So for those that you are not familiar, it's just like, how do you say this? Dimensionality reduction method, in which basically we are summarizing, so each of the dots in the plot is a cell, and we are trying to calculate distance between cells based on the expression of the genes, right? So cells that are more similar to each other should be closer to each other, and if they are fairly a bit more different, they should be a bit more different. So it kind of makes sense that this cell is very similar to this one, and a bit more different from this one, and super different from this one, kind of. And basically, what you can see is that here we have the 2 populations that we sequence, the stem cells, the neuroprogenitor cells, and using both gene counts or isoform counts, we can see the 2 populations. So here we have a bit more diversity, it's okay, we have more transcripts, so it makes sense. But the key thing is like, we are basically having all the information we had before, and on top of that, we can do something cooler. So that's the first step of how to see whether it makes sense or not. The second step would be to try to see if we can detect these changes in alternative polyadenylation, right? And this is an example that I'm showing you that it works. So I cannot be sure if it works always, but at least in some of the cases. So this will be the same plot as before. Here we have each dot is a cell, and here the cells are colored based on the expression of this gene, calmodulin-2. And this is what you would see when you look at the genes. But now that we are looking at the isoforms, what we can see is that, for instance, the difference in expression between the stem cells and the NPCs that is kind of more be clearly seen here in this violet plot. You can see that it's really because this isoform is basically not expressed in stem cells, while the other has a similar expression. And again, this is what you can see here. These two isoforms, so this one has a similar expression across cell types, but this one is exclusively expressed in the neural precursor cells, and that's what is driving this change in expression. On top of that, apart from having nice plots, we can experimentally validate this. So how did we do it? So this is the same gene that I have been showing all the time, just for the sake of it, so you only need to learn one gene. And this is, for instance, the kind of predictions that the Scalpel will give you. So if we would pool all the reads that we have assigned to the isoforms across these two cell types, we would see that what we see is that while in the NPCs, the two isoforms have similar usage, when we are looking at the iPS, we have one that is much more used than the other one. So how do we see this in the data? So if we do like a custom track in which basically we pile up all the reads that we have for a gene, what we can see is that these two isoforms that we are quantifying, the 204 and the 201, they have similar expression here in the NPCs, but we have one isoform that is much more abundant in the stem cells than the other. And experimentally, what we can do is do a PCR that basically is going to amplify a particular piece of gene, and then we can quantify how much of this gene or this isoform we have in different conditions. And yeah, I don't know whether you look at lots of electrophoresis, but basically here what you can see is like this isoform, this long one, and the short one. So the ratio between these two is smaller in the NPCs, what we see here, than in the iPS. And these bands never get better because this is related to the experimental technique. So we cannot get like the numbers as clear as this one, but believe me that this is pretty clear. So the program works, we can identify cell types that were fairly obvious, we can quantify isoforms, they are true, but can we use it in something a bit more relevant? Because maybe we want to see what I was telling you, what's happening across different cell types or doing differentiation, in our case, what's happening in Alzheimer's disease. So for that, we did a slightly more complex experiment in which basically when we took there were stem cells derived from control subjects or from Alzheimer's disease patients, and we differentiate them to generate neurons. And then what we did was to sequence cells at this stage, so between neural progenitors and neurons, so that we could do conventional single-cell transcriptomics analysis and also use a Scalpel group for looking at the alternate polyadenylation. And then with this data, basically, what you can do is what I was telling you before, you can compare the gene expression profile across cell types, or you can compare the cell proportions to see if there are any changes. And now on top, we can look at changes in alternate polyadenylation, right? So, okay, this is a great picture. I don't know much about how it was generated because I never go to the lab, but apparently I've been told that these are cells, and the things that you are coloring is the cells using antibodies that mark genes that are specific for different cell populations. And what you can see, well, maybe you cannot see. Now we can see better, thank you. But basically, there are not much difference between one and the other. So basically it's saying, if you would do conventional biology, you wouldn't know that there's anything that has changed at all. But when we do single-cell transcriptomics, we go from having these pictures that are fairly homogeneous and we are seeing the same, to actually realize that we have actually a diversity of cells. So we have cells that are dividing constantly. We have cells that are going to be giving rise to different regions, so to the neurons in different regions of the brain. We have cells that are giving rise to astroglial precursors that are the support cells in the brain. And also we have neuronal progenitors and different types of neurons in this data set. And you trust this, and you trust that actually these genes they are representative for each of these populations. At least, if you don't know the genes, I cannot explain you, but at least you can see that they are highlighting particular pieces of this plot so that they are marking different populations that we have. But the interesting is that in these populations, we also see this gene disruption polyadenylation. This is an example. Let's see if I can convince you that this is working. So this is another example as the one we saw before, the CDC42 gene. And in this gene, we have these three isoforms, this one longer, this one smaller, and this super mini isoform. So the ends are marked here with the green arrows. And I think that at least it's very easy to see that in all these conditions that are different cell populations, we all of them have a peak here, so we are using this polyadenylation site. But only in the neurons, we have this peak, that are these two isoforms. And this is how you will see when you look at how many cells, because we have the predictions per cell, right? So now we can look at how many cells are expressing this gene and what is the level of expression. And what you can see is that this isoform is massively expressed by most of the cells that we have, where basically none of the cells in the other conditions express this isoform. And not only that, but also it's expressed at the level of expression. The amount of mRNA copies that we have is much higher. This is another example, just to show that it's not that we always see the same, because it really depends on the gene. So in this case, we have the 4 cell populations that we had before, so replicating cells, astroglide precursors, let's say neuronal progenitors, so intermediate progenitors and neurons. And we are looking at a different gene, CAM3. We see again, this gene has 2 isoforms. And what we can see is that while the long isoform is heavily used for some strange reason in proliferating cells and in neurons, this other isoform, the shorter one, is much more used only in the progenitors. And again, this can be quantified not only in bulk, but also when we look at the specific cell populations. So this is great and expected, but now we want to push the method forward because these were still fairly easy cases. So the question is, can we find changes in alternative polyadenylation that are dependent on a biological condition? So it's not about the cell types, but in this case about having a disease. So this is the same plot that I was showing you before, but now I'm coloring the cells depending on whether they come from control patients or from Alzheimer's disease patients. And what you can see is that basically we have cells of the 2 samples almost everywhere. So we have all the cell populations, but the questions are there differences between the populations in these different groups? And at least I can tell you that there are differences in alternative polyadenylation. So for instance, this is another example that is YWHAZ, so it looks like a Polish one, right? So, but it's a gene, and then what we can see is that we have the Alzheimer and the control samples, and at least I think it's very obvious that this isoform, or I didn't say that these are astroglial precursors, so this isoform is heavily used in Alzheimer, what basically is not used at all in the control. So I think this is my last slide, so you can do something much more fun now. I hope that I have convinced you that single-cell transcriptomics is very cool, and we not only need to work looking at genes, but we can try to do much more and study, for instance, gene regulation, in our case, alternative polyadenylation. For doing that, unfortunately, or luckily, in single-cell there's still lots of work to be developed. So there's a lot of people that are developing tools for doing basically anything to think of, because most of them don't exist. And then we have developed Scalpel to quantify single-cell, to quantify alternative polyadenylation in single-cell. This data allow us to do classical single-cell analysis as any other single-cell data set would do, where it's great because you can use this data with any tool that has already been developed. And also on top of that, we can look at alternative polyadenylation. That would be the first attempt of looking at gene regulation and regulatory RNA regulatory mechanism in single cells. So just to thank the people in my lab, Franz basically is the one that was doing most of the computational work. and Anna was doing the cell culture and the single cell, but we also had Sandra helping with the validations, and also the people in the lab that contributed. The core facilities, bioinformatics core facilities, they are also really helpful. And that's all, and to you for your attention. And to say that we are looking for people for the lab, so if you're interested in any of these, please write and contact and come to Barcelona. Thank you.
We have a question? Yes?
Thank you for this really amazing talk.
You're welcome. Thanks for listening.
Now you showed us something that was already speculated, that isoforms play a really crucial specificity, especially in a cell type manner. Let's iterate this and showing us these amazing examples. I have actually two questions.
Okay.
So the first one, you showed us all the examples that are differing in these three prime and you showed us from neuronal cells, and I'm more interested to know what about RNAs or like genes that are isoform, that their isoform specificity comes from 5'N. For example, did you analyze data for LSD1, or maybe you know it as KDM1A, that has these two isoforms that are playing a crucial role in brain development?
So the answer is no. I haven't looked at it because the method is for 3'N, but one of the technologies that was more lately developed is doing the single cell and capturing the genes not from the ends. So I guess that you could use I think that you could use a similar principle, but just modeling distribution in the other way, and then you could be able to assign the reads to different isoforms that are changing in the 5'N. But I didn't thought about it. But now that you say it, maybe we should do that.
And my second question, if you would have unlimited access of money, of course,
Okay.
would you combine this data with single cell proteomics or single cell electrophoresis, for example, because we all know that sometimes mRNA isoform specificity does not necessarily represent in protein levels for the certain cells.
So yes, I would. So I would do much cooler things, right? So the first thing that I would do is, for instance, try to combine with a single cell attack that gives you regulatory regions, and maybe try to see how different regulatory regions are influencing 3' selection, or how the gene expression regulation and the RNA processing regulations are coordinated. Single cell transcriptomics, I think it's really cool, but we are still not really there yet. So what we can do is measure a few proteins that you have to define a priori and quantify them using antibodies. So I think that you cannot sequence the protein itself. I think that there are technologies being developed in this sense, but I think that we cannot afford them yet.
We didn't have for isoform specificity methods, but you developed, so I'm sure that for the proteins we can do the same.
Yeah, so it's being done. So you have another question?
No, this was my second question. Thank you very much.
You're welcome.
Yes?
Hi. I also like to talk. So I have at least one, maybe two questions, but what percent of isoform switching do you think could be detected with Scalpel? Therefore, like what percent of isoform switching do you really think is going to be targeted to the
Yeah. So honestly, I don't know, because we are doing the benchmarking, and the problem is that because we are doing the predictions at the cell level, it really depends on what's the depth of the sequencing that we have. So you have to think that for many genes in single cell, or let's say the less expressed 30% of your genes, maybe you are going to have one UMI in half of the cells, for instance. So if you only have that evidence, it's not that you can do a lot. So the idea of using all the reads coming from the same UMI for the EM algorithm is to take advantage of the fact that you don't only have one, but you have multiple reads. So you have multiple evidence, although you are only having an mRNA molecule. But what's the fraction? I don't know. So I think that if I would have unlimited resources and lots of data, we will be able to detect all of them. But I think that so far, maybe we can detect, I don't know, 10, 15% of the cases. And if so, I would be already quite happy. So it's not that bad.
Yeah.
We are detecting, for instance, in the case of the MPS, so the stem cells and the NPCs, I think that we detected about, I don't know, 200 or 300 genes with changes. We expect that there are supposed to be more, but there were changes, like clear changes driven only by alternate polyadenylation, so I'm okay with that.
So,
Yeah. Yeah, I have worked for lots of them with that.
And asking slightly based out of something just a but we've also got like a random single system tool where you can split the library. So having fragmented reads, you can also have someone who long reads, and then use this on your ask for nanoproteins. But then you would get the full length transcript. I wonder, would this still work if you had the full length full URL?
Yes,
Yeah,
it
and
would
then
still
you-
work because it doesn't care, but if you already have the full length, maybe you don't need this. You already have the isoforms.
Yeah,
Why would you need to quantify? The problem with combining single-cell transcriptomics with full-length RNA sequencing is that you get very few reads. So now maybe the data is not great, but I can be quantifying, I don't know, 10,000 isoforms in a cell. But if you do full-length RNA sequencing, maybe you go to 200, 300. So you have better data, but much less sensitivity. So I think that it's a compromise that you need. But maybe you could combine both.
Very nice talk. Am I right that your tool is limited to only non-isoforms and you cannot detect non-isoforms?
Yeah, so the tool is limited for that. The goal after we make this out, let's say, is to, so the problem is like for detecting new isoforms in single cell data, you would need much more coverage than what you have. So I think that the safe option would be to modify the annotation to include predictions that you could get, like using independent data or from independent experiments, and try to quantify those rather than try to do it using the single cell data itself.
Yeah, that's it. It's a very great tool. And another question is, what amount of reads do you take out during the internal priming filtering? Because you said that alternative identification site research is restricted mostly to 3' materials, but there are some cases where also the internal, intronic alternative identification. So how we go?
So all the reads that are in introns, we get rid of them. So just to make our
Or
life
the
easy.
amount, because in some cases
It really depends on the experiment. It really depends on the experiment. So in our data that was Droplet, we are working with cells and we have about 20% of the reads that come from introns. If you are working with other technologies or with other tissues, because it depends on how good you are separating your cells without damaging. So if you would work with 10x, I have been working with data sets that have up to 40%. So it really depends on the cells that you have. So that's one side. Then internal priming, maybe we are discarding, after discarding the internal, the intronic reads, maybe we are discarding like 10 or 20% of the data we have left. More or less.
Do you consider incorporating those reads in analysis and in the intronic identification?
So yeah, the idea is like the intronic polyA sites, the idea is to detect them by doing this thing that I was saying before of trying to expand the annotation, right? Because you can assign them to a particular transcript isoform and use this new annotation that you created, especially for the occasion, to quantify this usage. And that's particularly relevant, for instance, in cancer. And there are lots of these intronic events that are related to gene expression.
And the last question, have you tried to combine the alternative identification sites research with people they have?
No, because we cannot get this information in the single cell data, but there are very cool people that have already done it. And they have seen that there is a relation between the transcription start site and the polyA site, that the polyA site choice is related with the polyA tail length. So this has already been done in bulk. But we are still not there for the A length in the single cell. Maybe if we would combine with full length. No, the problem is like you're using an oligonucleotide, so we will have to change the technology to do it.
Thank you.
And there was one more question.
Yeah, thank you for the interesting talk. I think you almost answered my question already, but you can use this tool with different kinds of single cell RNA-seq data in general, so it doesn't matter how it's produced, or is there like a limitation?
So the program as it is implemented now, it works automatically with 10x and with Droplet. 10x is I would say, is the most popular single cell technology. For the others, we will need to modify basically, because the output that each of these programs produces is slightly different, so we will need to adapt it to work with these other technologies, and we haven't done it yet. So we have done it for these two technologies and it works well. Although the data is produced in a slightly different way and the results that you get are slightly different.
One more question. I think that's it. So thank you again, Mireya.